Hello, everyone, and thank you for joining us on this lovely Friday for another NeuroTools webinar. Today, I'm joined by Dr. Molly Bogue and Dr. Elissa Chesler. The content of today's webinar will be about the Mouse Phenome Database, an integrative database and analysis suite for curated empirical phenotype data. A little bit of information about our speakers. Dr. Molly Bogue is a research scientist at the Jackson Laboratory in Bar Harbor, Maine. She's the principal investigator of the Mouse Phenome Database project. Her interests include research replicability in preclinical research and sex differences. Dr. Elissa Chesler is currently an associate professor of bioinformatics and computational biology at the Jackson Laboratory in Bar Harbor, Maine. She holds a Doctor of Philosophy in Neuroscience from the University of Illinois at Urbana-Champaign. Her laboratory integrates quantitative genetics, bioinformatics, and behavioral science to understand and identify the biological basis for the relationships among behavioral traits. Without further ado, I'd like to hand the microphone over to Dr. Bogue and Dr. Chesler. Thank you again so much for joining us today.
Thank you, Devin, and thank you, Anita, for organizing this webinar series. The YouTube recordings of these webinars will be very useful for the research community. I appreciate the opportunity today to tell you a little bit about the Mouse Phenome Database 2.0. And I just want to say, Elissa, please jump in whenever you want to.
Will do.
Okay. So we'll get started. This is our homepage, our new homepage, I should say. It's MPD 2.0 has been live for only really a matter of months. So what I'm able to show you today is all new or relatively new. We'll come back to this website in a moment, but first I want to talk about the utility of MPD. So MPD allows users to access baseline and treatment data, choose mouse strains for various research applications, and I've listed a few there as you can scan through, accessing protocols, and accessing genotype data. On a more advanced level, it allows you to aggregate and analyze phenotype data, compare genetics results across populations, benchmark protocols in users' own laboratories, identify sensitized strain backgrounds for making new mouse models with genome editing technologies, assess replicability and reproducibility across experimental conditions and protocols, and assess sex differences and sex by genotype interactions. The current contents of the database, we've got phenotype and genotype data. The phenotype data is mostly per animal data, so it's primary experimental data. We've got thousands of parameters that are annotated to relevant ontologies. We've also got genotype data, including the Sanger data for 80 plus million genome-wide locations. And also we've got data for SNP data for the collaborative cross strains. Currently, there are over 1,800 strains having data in MPD, and those include inbred, recombinant inbred, chromosome substitution strains, F1 hybrids, transgenics, targeted mutations, collaborative cross strains, and diversity outbred mice. Just a quick MPD system slide just to make a few points here. Centers, large centers and investigators will contribute data and protocols, metadata into an area called intake and curation. And currently this intake and curation is basically curators manually curating the information, but what we're doing is developing an interface for data contributors to curate their own data. So this is a big project. In any case, they submit primary data, metadata, and it's entered into the database into an access controlled area that's password protected so that they can use QC tools, which we are also developing, to help inform them about whether to maybe delete data points if they're outliers, for example, and then re-upload and go through the same process until they're satisfied with their data and project, and then we can make it public. So MPD data and intake curation, that's all about data set being configured in standardized formats, ready to load into the database. Correct nomenclature is absolutely necessary. Also, a data dictionary needs to be compiled, which includes data type, description, units, treatment, other things. Ontology terms need to be assigned, and we use vertebrate trait, mammalian phenotype, and adult mouse anatomy ontologies. Experimental documentation has to be collected and formatted, and that gets turned into a protocol document, and animal environmental conditions. So data integration occurs at the strain nomenclature level, ontology term level, and also some protocol information is also used for integration. MPD documentation is based on RRID guidelines. This is going to become even more strict as we move into the curation interface, the new one that we're developing. But we're able to cover most of these items now. But we will also be covering research resource IDs where available. MPD projects consist of data, and this is an example of a data set that has 1 row per animal. And you can see that the columns are the measurements. Consists of metadata, for example, variable name, treatment, description, units, and ontology terms, and protocols. And we'll be able to look at a protocol in much more detail in just a minute. In addition to data sets, metadata, and protocols, we add a few other things just to add to the RRID guidelines. One is a descriptive title of the project, the contributing investigators, including the contact information, participants, primary publication, related publications, funding acknowledgments, and we also give it a project symbol so that it's actually easy to talk about in everyday language. One thing I want to mention before we take a dive into the database is our one-way and multi-way analyses. What we're going to see time after time in the demo is tables, results tables. And in the results tables, there's 2 types of rows. One is this type where you have a single term, time in open arms, and you have next to it a variable name. This is actually a single measurement. This is a single measurement, and it will go to a plot and analyses. This is in contrast to a situation where we've got a measurement that is composed of multiple measurements, for in this example, control and nicotine. So if you clicked on these down arrows, then you would get the component measurements. And again, these are individual measurements, and you can tell that because they have the variable name next to them. So hopefully this all makes sense when we take the demo, which I'll do now. Okay, here's the web page, the home page, as I've already shown you. Notice that we're looking at a preview node.
I'm sorry, Molly, would you bump up the resolution just a little bit so that I can see it a little bit better? Can you push the command and plus?
How's that? Is that better?
That's a lot better, thank you.
Okay. So we're looking at a preview so I can show you some unpublished data. But basically, the home page has a series of tabs, as you can see here. Most of them are self-explanatory. QTL Archive is a collection of mapping studies. The data is phenotype and genotype data. Data contributors, how to submit data, download center, and tools demo, which I recommend you take a look at if you'd like to learn more about our tools, because I'm unable to go through everything today. On pages other than the home page, we don't have the tabs. but we do have a menu pull down where you can get to each of these items plus a few additional items as well. So let's say we're interested in open field test data. You can see there's a type ahead, so it tells us what's in the database. We've got 2 examples of methods, an investigator's last name, ontology terms, and even a drug synonym. But what we're interested in is open field test, of course. Search results tells you basically what is contained in the database. Phenotype strain survey measures, the QTL archive, and phenotyping protocols. So let's go to the data. You can see we get these tables like I was mentioning earlier. All the data in here is open field. You can filter the results, you can sort results, and you can even search on columns if you'd like. There you'll see some check boxes to the left. Those have to do with working with the selected measurements, which we'll get into a little bit later. Right now, I want to be a little more basic. Data sets. This goes to a project page, where as you can see, we've got a detailed title of the project, publication, a short introduction with a summary of the procedures that were conducted. And you can see here that even more than open field test was tested. Contact information, including funding acknowledgements, etc. What downloads are available for the project. And what I really wanted to show you is the protocol. First, there's animal documentation, which shows you information about the environment of the animals, including feed and water, housing, etc. I'll just go back to the protocol. I just want to point out that a protocol can contain multiple procedures, as in this case here. But what we have is a workflow table which shows step by step what was done, the procedures, the treatments, the equipment, the data collected. Sometimes experiments are a little difficult to understand. So in this case, for example, we have added an image showing a schematic of the study design. Equipment, you can see that we include the vendors plus dimensions when available, reagents and supplies and solutions, and then the actual procedures are listed. As you can see here, they're quite detailed. And at the end, we even show what some of the apparatus look like in case people are unfamiliar with them. For example, the third one, I was definitely not familiar with that one. Below that is data collected by the investigator. And with our new new curation interface, we will be expanding this greatly to include really a table, more like a data dictionary. Definitions are below, and then references. So you can see we've got quite a lot of information for each protocol. Now if I just back up 2 pages, because I want to get back to my open field test result. Let's say, for example, well, let me just also mention this. The procedure link goes directly to the procedure. You don't have to go through the project. You can go directly to the procedure anytime you are near the measurement name, which is here. Panel, number of strains, sex, age, sample size, year. For this set, we go all the way from 1993 to 2018, so there's quite a lot of data there. Let's say, for example, that we're interested in collaborative cross strains. This is the data that I wanted to show you. Here's an example of multiple measurements being in a particular plot. You can see that we've got first 5 minutes, second 5 minutes, third, etc. So I'm going to click on this and then come back to the single measurement plots in a minute. But this is the multiple measurement plots. You can see we have information at the top, including a list of strains if you wanted to see what they are. Again, the procedure is one click away. Ontology mappings. We have options for plotting above the plot. We're showing female and male. And you can see there's quite a range of phenotypic differences in this particular measurement. Some strains don't react much at all. Others have very high activity and remain high. Others are hyperactive and decrease almost exponentially, but they're still very high at the end at 5 minutes. So there's really very interesting phenotypes in here. If we scroll down, we get to a bar showing individual measures. If we click on one of those, we get the individual measurement for the first 5 minutes. Again, there's information at the top. Plotting options, we can hide the individual animals, for example. And then we have the analysis information. For example, the measurement summary, which is the number of strains, the mean, median, standard deviation, etc. We have an ANOVA summary at the top, a table. You can see that strain has an effect, and strain by sex also has a bit of an effect. Below that is the QQ plot, where we're plotting theoretical versus observed quantile values. And you can see that this particular measurement does follow a fairly normal distribution, although it's not exact. You can mouse over these points and get the animal ID in case you want to track down that particular animal to see if maybe something was wrong with it, or maybe it should be taken out of the data set. Strain means statistics. The typical statistics, you can sort these, so I'm able to quickly see what the outlier strains are just by clicking on Z-score. You can also click on the mean and get to individual animal data. And this is listed by sex by Z-score, so again, you can see what some of the outliers may look like. And then least squares means, and I won't show those right now. So those are the basic plots and analyses that we have for the data. For each measurement gets these analyses. What I want to move to now is, actually I'm going to move to the live page. A lot of people look for strain information. They may want to know what's the baseline for a particular parameter. You can click on strains and get to a listing. You can list them by specific panels or strain types, for example. But let's just pick a strain, because most people come in with a strain in mind. We'll pick BTBR. You see, we've got an image of the mouse, a link to the vendor, and we link to Taconic, Charles River, Harlan, any other vendor that we have information for, so it's not just JAX. But you can list all studies involving the strain. You can find the phenotypes where the strain is an outlier, download all measured phenotypes for the strain, retrieve SNPs for the strain, and compare the strain with one other strain. And if I know exactly what I'm interested in, I probably don't want to list all the studies, and I probably don't want to down all the measured phenotypes. That's why we have this prominent search box. We're hoping users use that instead of trying to browse the data. So let's say, for example, we're interested in novelty. We get 43 results. And let's say further than that, we're interested in the whole board test. We can click on one of these down arrows and get to the BTBR data. If you mouse over, you get the standard deviation and the z-score for both of these. But what you probably want to do as an investigator is take a look at the data itself. And you can see that BTBR actually is very average for this. I think what I would like to do now is just show you how you can add this to your collection. So I'm going to add this measurement to my collection. I'm going to put it in a folder, and that folder I'm going to call NIF Webinar, and just add it. And now it's in my collection, and I will come back to that collection in a couple of minutes. Another thing you can do is actually find outliers, which is very powerful. And also you can compare this strain to another strain, which we'll look at really quickly. Let's say you're looking for AKR and notice that suggestions pop up, and it's extremely important to get the nomenclature correct. So this is really helpful to be able to do that. Compare phenotypes or compare genotypes. We'll take a look at phenotypes. And you can see that we've got 395 different measurements to look through. So that's quite a bit. So what we would probably want to do is make this a little more stringent. We can do that by changing the p-value, and we come up with 56 measurements. And let's say we're interested in, let's say, tail suspension test. You click on that, and then we can see what our values are. Again, you're going to want to look at the actual plot itself. AKR is here, and BTBR is way down here. So you can see that they are quite different. Okay, let's take a look now at my collection, which is called My MPD, and take a look at more advanced applications. This is the measurement that we just added a couple of minutes ago, but you'll notice that we've got measurements from multiple projects. It doesn't have to be the same project, from multiple different methods. So we've also got behavior data. We've also got, in this example, we've got morphology data. So you can combine all sorts of data into your collection. What we'll do is select all boxes, work with selected measurements, and you can notice below that you can actually manage your measurements in your collection if you want to. Right now we're going to use the visualization analysis tools. And I'm just going to open this up so that we can see all the measurements. And what you'll notice is that instead of these measurements being listed in a series, now they're listed singly. That's so that you can deselect what you don't want. So I'm just going to go through and deselect some of these. And we've ended up here with 7 measurements. And you can see across the top, the eligible measurements are in this sort of blue color. The grayed out one indicates that it's not eligible because we have more than 2 measures. But let's take a look at scatter plots and correlations. You can see the measurements are listed at the top. We could expand this, but we don't need to at the moment. Again, there's plot options. We can look at Spearman or Pearson. This is a typical correlation matrix where the correlation coefficients are either red or blue, different shades of red and blue depending on whether it's positive or negative. Positive is blue, of course. And the size of the circle is related to the p-value. So the larger the circle, the smaller the p-value. I can mouse over any of these cells and get information, including more information about the individual measurement. So I could click on that and get to the individual measurement. But I want to really click on a cell and take a look at the scatter plot a little more closely and get additional information, more data. So we've got the Pearson and Spearman R and P values for both sexes, for females and for males only. Again, there's options for plotting. We can show the point labels. And below, you can even highlight strains if you want. And of course, mousing over will also get you the identification of that strain. This table is downloadable, as well as the plots, which I don't think I mentioned before. All the plots are downloadable into publication-ready images, and we encourage people to do that. Okay, I'm just going to close this window, come back to our tools. Again, there's too many tools really to go over today, but I just want to show you that if I pick another tool here and watch this here, because what I'm going to do is deselect in my list, and you can see that it updates immediately. So my tools are a reflection of what's been selected up above in the table. Another thing that you can do is tile these so that they're side by side, in case there's some reason that you would want to look at them side by side. Each panel has its own controls, so you can enlarge or move around or close, which we'll do. We'll close those. One other tool I do want to show is find strains by criteria fit. Let's just expand that. And this is based on z-scores. You can control the level of z-score for each. High-end outliers are in yellow, low-end outliers are in purple. And I like to show the z-scores so I can see them. So what we're going to do basically is, for each measurement, choose either high, medium, low, or show, depending on what your criteria are. So as you can see right now, the strains are just listed by alphabetical order. But as I start to apply criteria, you'll see that you immediately get a change. So C58 right now is the top choice based on this one selection. If I want to do thigmotaxis at low level, you get a change. So we can do all of these. And what we end up with then are the top choices for matching these criteria. And if you look closely, you can see that NOD and FBB both meet the criteria, but NOD is actually more extreme than FBB. So that's the strain by sex that you would probably want to use for your application. So this is particularly powerful for choosing strains for genome editing, for example, or just trying to develop new mouse models for human disease. Okay, we'll close that. So what I've shown you is integration by strain, integration by method. You can also do integration by intervention, such as cocaine, or you can do ontology terms. So again, here's all the depression-related behavior traits. I could select the boxes, and I can work with these measurements directly, or I can put them into my folder. Either way, if you put them in a folder, they're persistent, and you can come back to them. So how much time do we have?
We are at 11:30, 11:29.
Okay. I think I better skip. I think I'm going to skip the genotype part of this and go back to the slides, just so we're sure to have time for discussion. What I want to show you now are just some things that we're involved in that are under development. I've mentioned several times now this curation interface. We're calling it the curation dashboard. It's under development. Multivariate tools are under development, particularly the multivariate outlier detection. We're developing a mutant controlled data analysis and QC platform, and this is primarily for KOMP type data. We have a new collaboration with Yoav Benjamini at Tel Aviv University, and the basic idea is to take MPD data, for example, open field test data, total distance traveled. Let's say we collected that from multiple studies across multiple genotypes and came up with a matrix. And using this matrix, we could compute a genotype by laboratory estimate and use that estimate in a single lab equation to see if those results are likely to be replicable or not. We're also doing a collaboration with Rob Williams at University of Tennessee Health Science Center. And the basic idea is to exchange data. They have a lot of data that we don't have, and we have a lot of data that they don't have. So the idea is to do some data exchange so that we both have essentially the same data and provide ways of getting back and forth between our respective resources as seamlessly as possible, so that users from MPD, for example, can use gene network tools and vice versa. And gene network users can get immediate access to protocols and the per animal data. We also have a collaboration with Elazar Eskin at UCLA, genome-wide mapping using PyLim that the ZAR lab has developed. QTL meta-analysis also with PyLim and Metasoft data analysis. So we welcome collaborations to populate the database and to develop new tools. If you have data to submit, please check out the data submission guidelines. They're available, and if you have questions, please email us. At the bottom of each page in the website is a footer that has help and feedback link. Please use that if you have some feedback to give us or need help. Or you can email us directly at phenome@jax.org. And this is the MPD team. You can see that we have a lot of support from the Computational Sciences group at the Jackson Laboratory. So I just wanted to point that out. And of course, our funding from NIDA is primary source, and also we get some funding from NIA. And with that, I'll stop.
Excellent. Thank you, Dr. Bogue. We do have a few questions lined up for you.
Let me start.
Sure.
Can I start with one, unless there's somebody else who wants to?
You have the floor, Dr. Bentos.
Okay. All right. So first of all, thank you so much. This has come a really, really long way, and it's such an important resource. The first thing I kind of wanted to maybe get you to elaborate a little bit on, because I know that you know really well what the collaborative cross is, and what KOMP is, and what have you, but there are lots of people who still don't know what those resources are. Would you just mind taking a couple of minutes and really explain what are these resources and why they're important to researchers, out in the wild?
I think Elissa probably could answer that really well. Elissa, do you want to?
Sure, I'll take a crack at it. You know, the collaborative cross is a mouse population that was designed by members of the Complex Trait Consortium for probably around the millennium, and the effort began with a multi-site breeding operation. And what we've developed is a systematic intercross of 8 inbred strains, 5 of which are common, widely used strains, including B6 and AJ and 129. We also included wild-derived strains, PWK, WSB, Castaneus, NZO is another strain from the common set. And that mixture of common and wild-derived strains segregates most of the mouse polymorphisms that are out there. And so what that allows us to do is survey all of mouse genetic variability. To make the strains, those 8 founders were intercrossed randomly, or rather in a planned fashion, and then inbred to create what are called recombinant inbred strains. In a recombinant inbred strain, we've recombined the genomes of the founders, but then by re-inbreeding them, we create isogenic strains where every member of the strain is identical, but each member of each strain is distinct. When people characterize these strains, they have genetic randomization of the recombined genomes of the founders. And so things like the assumptions of independence that drive correlation analyses that are so predominant in the Phenome Database are unbiased by the relationships among the strains. The existing historical set of inbred strains was subject to a genealogy analysis at JAX almost 2 decades ago now. And we know from historical records that all of the inbred strains have many different relationships among them that make them non-uniform. Thus, the correlations violate some of these assumptions of independence. So the collaborative cross gives us unprecedented genetic diversity in a genetically well-randomized and unbiased population. The diversity outbred population intercrossed the collaborative cross strains, and that gives us a very precise set of recombinations in a population of unlimited size, which randomly recombines the genetic material of the 8 founders. It allows for extremely precise genetic mapping analyses and systems genetics analyses, but unlike a reference population, the recombinant inbred strains, every member of the diversity outbred population is unique and needs to be genotyped and phenotyped for genetic analysis. And you can never repeat a study in precisely the same genotypes again. So, the knockout mouse population is another one that Molly talked about. The KOMP program is part of the International Mouse Phenotyping Consortium and International Mouse Knockout Consortium. This is a global effort to make a gene deletion mutation for most of the genes in the mouse genome. And each of those are characterized across multiple centers in a standardized set of phenotyping procedures. And we've represented all of the data in the Phenome Database to allow for new integrative and interactive analyses. Most of the data from all of the centers is available at mousephenotypes.org, and we may be able to pull in increasing amounts of that data in the future.
Thank you so much. I think this is really important because one of the things that I don't think goes into enough people's protocols when they say, hey, I wanted to try this protocol on a mouse, they don't really know how to find the right mouse. And this is just an absolutely tremendous resource for that question. So I want to be as loud about this as possible. Because
yeah, we think one of the really neat applications right now with the strain finder tool, a lot of people want to make mouse mutations on a genetic background that's either sensitive or resistant to a particular phenotype. And you can use this tool and those data with the advances of CRISPR, which allow much more facile genetic engineering on diverse backgrounds. You can select the most appropriate backgrounds or backgrounds in which to study your mutation.
Yeah, and I would also would love to plug the fact that, yeah, there, which actually Molly had already pointed out, if researchers are, they get through a certain part and then they're a little confused, the resources at Jackson Labs are amazing, and you guys really know what you're doing with these things. So asking a question is probably better than, hate to plug your system of questions up, but I mean, these things are so absolutely critical. And then the second sort of set of things that I would want to just kind of make sure that everybody understands going into this. Can you speak a little bit more about, submit your data? Because one of the other things is, like, this is such an important resource to be able to have all of these standard measurements on top of every one of these strains, right? But you're never going to get everything, because that's just the nature of the beast, right? So there are going to be other people who have done various tests on some subpopulation of these mice. And, the submit your data, can you talk about, what are the things that are involved with submission? I know you have a very deep and precise curation workflow. What can the investigator do?
What we're trying to do now, what we're trying to do now is to make it easier for experimentalists to prepare their data for submission, to bring it as far along as they're able. And Molly can describe those curation tools. That will enable us to more readily integrate their data into the system and to expose it to the analysis tools, and also for other investigators to access it and learn from it. The detailed protocols are an essential part of this. It takes a lot of work to self-curate, but we think that that's going to give us the most precise and informative descriptions of these studies.
All right. Anita, I saw another question you had in regards to the statistics visualization. Would you?
I have so many questions.
Well, yeah. Would you like to voice that, or would you like me to kind of paraphrase it?
I think if there's anybody else on that would like to know more about the protocols and other things, because, I'm sure that there are some voices. For example, Tom.
Yes, so I want to apologize for arriving late. I was on my bike. So if this has been covered, just tell me it's been covered, and I'll watch the webinar when it's posted. So I'm sitting here looking at this lovely phenome.jax.org/strains, and this seems like it answers a question that I've had for a very long time, which is, how do I identify a mouse strain? And not only that, but it actually it has numbers for them, which are machine understandable. So my question is, when we look at these strains like a C57 Black 6J or an A/J, how do you think about them conceptually? Are they a known genotyping result? Are they a breeding record history? Are they, what is a strain?
Well, a mouse strain, an inbred mouse strain, is very well characterized here, especially those that have the /J. We have genetic quality control programs in place to ensure that those strains have stable genetics across many generations, and the data collected in those strains as indicated, should be comparable across multiple years and multiple studies. There are spontaneous mutations that emerge in a strain. Some of those have been reported, discovered, and characterized to great effect. But we also know that by and large there's stability. In order to integrate data, we sometimes have to correlate across strains, and to do that a little bit more loosely. But the strains themselves are very rigorously documented. People append sometimes a laboratory code to the end of a strain name. That indicates that it's been in their own laboratory for more than 9 generations. And at that point, we think that it's genetically deviated from the original stock. And so using those lab codes is a very important means of identifying whether or not there's potential spontaneous mutations that may be separating that strain from any other with the same strain name.
And if you wanted to determine whether or not it had actually drifted, you would have to go through the battery of tests again?
One of the important things that you can do, well, phenotypic drift, yes, you'd have to confirm the phenotype, but increasingly there's a lot of genetic and genotypic data available for these strains, and it's becoming more affordable to genotype and resequence strains to determine whether or not there's major or even minor differences that have arisen over time.
Mm-hmm. Mm-hmm. So you could in fact think, tying into the protocols question, you could in fact think of a strain as being defined by a very well-defined protocol for identifying and classifying members that fit the definition.
There is a strain nomenclature group in the Mouse Phenome Database, and they apply some very strict rules for strains, substrains, lab codes, and a number of other attributes that define rigorously what a strain is. And so you might actually, if you're interested in this topic, learn more about it from the Genome Database.
Okay, perfect. Thank you very much.
Okay, so now I'm going to have to applaud you for adding RIDs because, you know, I like those. But also, you know, if you want to take anything offline in terms of, do you know how to link it to them and all of that? If anything's not well enough documented, please let me know. And even, you know, it would be great to understand if there are things that you guys would need in terms of identifying tools and reagents that, you know, I would love to put those into a list of things that, you know, would be useful to the community. So do you want to talk a little tiny bit about that, or are we good? Should we take it offline?
It'd be useful to just say, you know, it's a heroic effort to curate data like this for the community, and in some cases, people developing some of these terminologies and extremely important to making that curation effort effective. We'd really welcome it if you wanted to scan these protocols, or even if you developed a system to scan the protocols to look for words that may have RRID terms that we're missing, and also to help us integrate the protocols better with the RRID resources.
That would be actually a fantastic use case for a project that we're starting to embark upon. So I will most definitely take you up on that offer.
Yeah,
excellent.
And I also on related to map the protocols and mapping to ontologies. These are great. I will take a look through all the protocols that we have here and see how we can map them into our methodology work as part of the NIF ontology as well, so that people can find those protocols and make use of them.
That's great. One of the important foundations of the replicability collaboration with Dr. Benjamini is that we're wanting people to be able to search for a collection of studies using similar approaches so that they can estimate how their results might be influenced by genotype and laboratory environment specific effects, and whether their result is expected to replicate across laboratories. And having controlled terminologies for elements of the laboratory environment associated with these protocols is going to be an important way in which people can find those similar studies.
Mm-hmm. Yep. Yeah, this is great.
No, agreed. And from my side of this, the RRIDs just help you do that based on reagents, but there are so many different levels to being able to actually compare studies based on things other than something very superficial like a mesh term. There's so much deep, great data in there that can be compared, and you have just pulled out, in this resource, you've pulled out so much of it that I'd love to be able to make sure that as many people know about this as possible, because it can be so helpful, for relatively kind of benign things. I want to study feeding in a mouse, and which mouse strain should I choose, and how should I do that? Which ones would be more responsive if I want to start knocking out genes? This is just, one of the first things you need to do is figure out, okay, what's the average for this, for my control? And if you can look at that and compare it to the strain average numbers immediately, it's really super helpful to know that everything in your lab is actually happening in a way that it should, compare, because you've got a really big sample size for a lot of these. So it's just tremendous. And thank you so much for doing all this work. This is heroic. This is absolutely heroic.
I'm looking at the various results for protocols and thinking, oh, this is what I thought would take us 10 years to be able to do, but you guys have done it. So
thank you. Yeah, it's been only 15 years for Molly. My other world that I inhabit is that I actually have an experimental component. And researchers in collecting data, or we want to bring a new procedure into our lab, we'll actually go to the Phenome Database, choose a protocol.
You're fading out.
Yeah, we're losing you a little bit, Dr. Chesler.
Okay. Where did you lose me? One of the things I do in my laboratory when I have new technicians learning a procedure is to have them go to the Phenome Database and implement one of the procedures that's in there, and then compare their experimental results on known strain differences to what's been previously reported in the database. And so it's an important resource for us in implementing new procedures.
And that actually sounds like it's not just for implementing new procedures. It's actually for train. It's also a training resource in some sense.
That's right.
Excellent. Well, Tom, Anita, do you have any more questions for our speakers?
Oh yes, I'm sure that I do.
Yeah, I'm sure we could carry this out for quite a while.
Yeah, but
Well, I think we could. It would have to involve some beverages.
Yeah.
But so thank you too very much for telling us about this, and I think Devin's gonna put this up on YouTube. But I personally enjoyed this webinar so much.
Thank you.
Yeah, thank you.
Yeah, this was absolutely excellent. We really do thank you for taking time out of your Friday to join us and present. I will have this on YouTube in probably an hour or two, just after I make a few edits. It'll be up on our channel, and I will send you the link, and I'll also put the link on our new info webinar series website, the NeuroTools Webinar website, which I have just put into the group chat. Dr. Chesler and Dr. Bogue, is there anything else you'd like to add?
Not for me.
Nope. Thank you all very much for the opportunity. And if you have any data to submit or feature requests or feedback, please do send them to Molly and I. Thanks.
Yeah, thank you very much.
thank you all.
Excellent. Thank you again. And I'd just like to give a short plug for our next webinar. The details are on our upcoming webinars page, which I've posted into the group chat. I love this title. It will be the Red Queen's Race, How Transparency, Interactivity, and Gamification Can Be Used to Improve Data Quality. It will be presented by Dr. Hawk Barch at 11 a.m. Pacific Standard Time on May 4th, 2018. For further details, please feel free to visit our website. Thank you again to Dr. Bogue and Dr. Chesler for joining us today. And once again, this link will be on YouTube in a few hours. All right, I'm going to end the webinar now. Thank you again so much.
Thank you.
All right, bye-bye.